Okay, so for question number 1, it asks us to talk about how solutes and water are reabsorbed in the kidney tubules. So we'll go ahead and draw a little picture here. This is going to be what we're talking about here. We're going to have, here we have the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and then down to the collecting duct. So this is kind of our reference picture right here. And so what I'll do is I'll draw different kidney cells and organize them. There's going to be 6 different kidney cells that we talk about that are going to cover each type of reabsorption. So I'll go ahead and draw those now. So now we're going to talk about each section of the nephron here. So here we have number 1, which will be right here, which is the proximal convoluted tubule. Okay, so first here in the proximal convoluted tubule, we have different types of transporters here, different types of things that are going to be moving in and out of the cell. So first, we have 2 symport channels right here that move both sodium and glucose, as well as amino acids and sodium. And so 100% of the glucose gets transported into kidney cells, but only about 70% of sodium gets transported in. And so here we have this opening where water can go ahead and pass through and get reabsorbed back to the blood. So this is our water. And also what we have here is a passage in between 2 cells that is open that water can also pass through. What we have that is being brought in as well with this water is calcium and potassium that is also being brought through back into the blood, which is what we call drag. Here on the other side of these cells, we have another type of transporter that is able to bring glucose back into the blood. And on all cells, we have the sodium-potassium ATPase pump. So we have potassium into the cell and 3 sodium out of the cell back into the blood as well. So these are the main components of the proximal convoluted tubule and how the reabsorption happens in this specific section. So now going back to our drawing of the nephron here in this straight section, we're going to have our second cell. This is going to be the straight portion of the proximal convoluted tubule. So in this section, we have 2 different interesting transporters that are antiport. We have a chlorine and H+ transporter, and we also have a sodium and anion transporter. So what happens here on the outside of the cell is this anion will combine with this hydrogen atom to come back into the cell like this, and then they will separate again into hydrogen and anion. So that will allow this cycle to keep going where the anion and hydrogen ion will combine to enter into the cell, and once they're into the cell, they will split in order to keep this cycle going. And so here we have, in result of that, a chlorine channel that allows the chlorine to return back to the blood, and that is what we have in the straight portion of the proximal convoluted tubule. Our next section here will be number 3, and this is our descending section. And the descending section is pretty straightforward. There's not a lot that is going on here except for the reabsorption of water through the cells back into the blood. So now for number 4, here we have the ascending section. And this section has a few different things going on. So we have a 3-way transport here of sodium, potassium, and 2 chloride ions coming into the cell like this. And so here we have our continuous sodium-potassium pump that is able to regulate the sodium going back into the cell. And just like before, we have this chloride channel that allows the chlorine to go back to the cell, and we also have some potassium leak channels that allow the potassium to return back into the blood as well. Something else about this ascending section is that there are proteins that will block off this transport here, the area between the cells, so that the water that is trying to come in is not able to be reabsorbed because it had been reabsorbed in the proximal convoluted tubule and descending section. So back to our drawing, we have number 5, which is the macula densa. Here in the macula densa, we have a transporter that is bringing in both sodium and chloride, and so we will have the same thing happening here. The sodium will go back to the blood through this sodium-potassium pump, and we will have the chloride channels allowing the chloride to return back to the blood as well. And this section is still blocked off to water. And here our last section, number 6, we have the collecting duct, which is where we have open channels for sodium as well as potassium being able to leave the cell. And here, once again, we have our sodium-potassium ATPase pump and yet another potassium channel to return back to the blood. And we have the area between the cells also blocked off after that water absorption has been done. So now when talking about the idea of threshold, that is the idea where each one of these transporters along these cells here can only contain a certain amount of these ions traveling in and out of the kidney cells. So say, for example, glucose, we have a certain amount of transporters on our kidney cells that is accountable for glucose. And if there is too much glucose going into the kidney cells and the kidneys are not able to filter all of the glucose, then that glucose will continue into our urine because it will not be reabsorbed into the blood, and that will let us know that there's an increased saturation of glucose, and that will cause some problems. It will let us know that our transporters are either not functioning well or we have too much glucose in our body. Okay, so for the second part of this question, it asks us to talk about filtration. So what I've done here is I've taken this little Bowman's capsule and kind of blown it up to kind of show a little bit about what this filtration is all about. So this is the afferent arteriole here and the efferent arteriole here. And so as the blood is traveling into the Bowman's capsule, here we have all of these tubes, this little section where the blood runs through and gets filtered. So if I were to take this little section here and blow it up, we would see a few different layers. So below here we would have, got like a basement membrane here. We have capillaries as well as a fenestrated endothelium. And so the thing about filtration here in the Bowman's capsule is that it is based on two things. It is based on size and charge. And so unlike the kidney cells that are specific with transporters, filtration just happens based on how big the molecules are and the charge that they have on them. So as the blood flows through these tubes here, depending on the size and depending on the room here in between these membranes and these fenestrated endothelium, we will have these small particles be able to be filtered out of the blood and be brought down into the rest of the nephron. So the filtration is simply based on how big the molecules are and the charge that they have on them. As blood flows through the Bowman's capsule, it will filter out these different kinds of ions based on the pressure. And so what we have as well is a number that is called the GFR, which is the glomerular filtration rate, which on average is about 125. And that is the rate at which the blood that is flowing through the Bowman's capsule gets filtered and returned back to the body. So with blood flow into the Bowman's capsule, there is a certain pressure associated with it. So on average, we have a pressure of 55 millimeters of mercury that is entering through the afferent arteriole. And so what we know is that there are macula-densa cells along the nephron that can sense certain levels of ions in the nephron that has been filtered. And so what can happen is there can either be vasoconstriction or dilation of the afferent arteriole in order to regulate those levels, and which will ultimately change the GFR. So say we have a change in our sodium chloride levels that are detected by macula-densa cells that will cause a chain reaction that will lead to either a vasoconstriction or dilation that will then cause an increase in GFR or a decrease in GFR. So when the afferent arteriole is dilated, there will be more blood going to Bowman's capsule, which will cause an increase in GFR. And here there will be less, which will cause a decrease in GFR. So now when we talk about the idea of clearance, this is the equation that clearance of a certain substance X is equal to the urine concentration of substance X times the urine volume rate over the plasma concentration of a certain substance. And so we know that clearance can be equal to the GFR if there are certain assumptions made. So we know that clearance is equal to GFR if we know that there is no reabsorption of X. We know that it is freely filtered. And we know that there is no secretion.